Okay, welcome everyone. We're so glad you decided to join us. We'll be exploring antibiotic resistance in wastewater systems today. My name is Lori Krings. I'll be moderating. We are coming to you from the Center for Technology and Training at Michigan Tech in Houghton, Michigan. And I'm joined by a great team. We have Daryl Gotham, who'll give you some background on the Environmental Finance Center network here shortly, and Ingrid Sandberg, who's keeping things running smoothly on the IT side. Thank you, Ingrid. And we're thrilled to have Ishi Keenum with us today as our instructor. She's an assistant professor here at Michigan Tech in environmental engineering, and we're really looking forward to what she has to share today. A couple quick things to point out as we get started. You are invited to put questions into the Q&A or in the chat, and I will be monitoring both areas. We'll have a Q&A session at the end. And we are recording today's session, and we'll send the recording link to you when certificates go out. And I will be dropping the slides into the chat here shortly as well, so hang tight for that. So if you do need a certificate of attendance, if we could go to slide 2, we'll talk about the things that you need to do that. So if you do need a certificate of attendance for today, just make sure that you follow the steps here on this slide. The biggest thing to point out is that you have to be pre-registered and that you need to complete a SurveyMonkey link that I will give to you at the end of today. I'll drop that into the chat. And if you happen to be watching in a group, which is fine, just know that you'll have to have everybody do that SurveyMonkey link. Okay, and we will get you those certificates within 10 business days. But if you do have any questions, you're welcome to email us at that ctt@mtu.edu address. Okay, so then Daryl, if you could please tell us more about the Environmental Finance Center Network.
Thank you, Lori. This presentation is brought to you by the Great Lakes Environmental Infrastructure Center through grant funding received by the Environmental Finance Center Network. The Great Lakes Environmental Infrastructure Center serves small communities in EPA Region 5, providing training, research, and technical assistance to water and wastewater utilities. I'll turn it over to Ishi now to begin her presentation.
All right. Hi, everyone. I hope you can see me. Please, if you have any technical issues, put it in the chat and Ingrid will figure it out for us. But today, I'm an assistant professor. My name is Ishi Keenum, and I'm an assistant professor in environmental engineering at Michigan Tech University. And today I'm going to talk about research that I do on antibiotic resistance in wastewater systems, looking at kind of the state of the science and why do we think about this in wastewater, and then going into ways we can test for antibiotic resistance, and then the role of mitigate wastewater treatment and mitigation strategies to reduce antibiotic resistance spread in wastewater. My email is here on the slide in case you all want to reach out at any point in the future. I love talking to people, so would be happy to talk in the future. All right, so the objectives for this seminar, we're going to explain sources and pathways of antibiotic resistance in wastewater systems. We're going to discuss the role of wastewater treatment plants in the spread and control of antibiotic resistance. We're going to describe testing methods for antibiotic resistance genes and bacteria. And we're going to evaluate mitigation strategies to reduce AR through wastewater treatment, like what treatments work or what do we know right now. There will be a Q&A at the end of the webinar, so feel free. You can put questions, I think, in the chat or in the Q&A box, and we will address those at the end of the webinar. Okay, so what is antibiotic resistance? So here I've kind of got a lot of headlines that aren't really intended to scare you, but I want to make sure that we all are kind of on the same page about what we're talking about here and why are we talking about this? So as you can see here, I guess if you've all heard of the super bugs, or they get in the news on these, basically antibiotic resistant bacteria are often thought of as super bugs because they are bacteria that we historically have been able to treat with antibiotics. But because of kind of repeated use of antibiotics, we are finding that bacteria are evolving to survive the treatments that we need. And so we can think about antibiotic resistance has been recognized by the US CDC and the World Health Organization as a global health threat because antibiotics are becoming less effective as bacteria evolve ways to survive treatment. This is, I mean, quite honestly, it's pretty scary. Antibiotics have been one of our primary ways to treat infections over the past 90 years or so. And antibiotic resistant bacteria are particularly scary because unlike many infectious diseases, they can affect individuals at any stage of life. So just kind of participants on the call, maybe turn to your neighbor, or if you're in a group, just think about if you've ever taken antibiotics for any reason. And I guess you're still attending, so it means they worked, which is really what we're trying to do. We're trying to preserve the efficacy of these really, really valuable drugs because we haven't done a great job of making new antibiotics. And that's really been a challenge, is these bacteria are evolving resistance faster than we can develop new antibiotics for them. Since 2019, 9 of the 19 antibiotic resistant bacteria that are monitored by the CDC have increased in prevalence, meaning they're more likely to occur. And it's estimated that's currently around 1.2 million deaths globally per year. In the United States, as of the CDC 2019 report, what actually came out in 2022, anyway, there was about 2.8 million infections per year in the US. And that led to 35,000 deaths per year. And in the EU, it's fairly similar, 33,000 deaths per year. So that's kind of the big intro and like a little bit on why we care, why we're doing what we're doing, and a little bit the motivation. And then I think it's really important that we think about the role of wastewater treatment and critical infrastructure in infectious disease and why it's so important. So here's kind of one of the timelines we think about the biggest advances in public health. So a lot of those, I know this is a wastewater webinar, but we know about the first use of chlorination in drinking water systems in 1908, contributed to the greatest the greatest life-saving intervention that we've done yet as a species. After that, we started to get better at surveilling foodborne outbreaks. And then we even better yet in around 1940. So those were the first chlorination. 1940, we developed the first use of penicillin, which was our first antibiotic. And that really, really changed the game on infectious disease and how people surviving basically when they get these deadly infections. One of the things that I do want to note, and it's not on the graph, is that by 1942, actually we had our first antibiotic resistant case. And so there's documented scientific articles going back that far that we knew antibiotic resistant bacteria were evolving just as we were developing new antibiotics. So we know antibiotic resistance is a problem. We know wastewater and water treatment are critical infrastructure that save lives, but how do they connect, right? Like let's connect some dots. So I think it's really important that we think about antimicrobials. We use antimicrobials outside of just human medicine. So this is why I think about it as an environmental engineer. We use antimicrobials in veterinary medicine and for our dogs and cats. So for veterinary medicine, this could be for an aquaculture. Sometimes it's for maintaining school health, but that practice is decreasing. In dairy cattle, we'll often use antibiotics if a cow has mastitis. It's a kind of a common intervention. For chickens, historically, it was used to maintain herd health because they're often living in pretty tight conditions, but actually consumer pushback has radically changed that in the last few years. And then I have a dog. I love my dog, Monty, but we do use antibiotics quite a lot in like companion animals as well. And we think about those a lot because people interact really closely with those. I cuddle my dog. We also use antibiotics when we think about the environment, right? So if we're thinking about where we make antibiotics, pharmaceutical production facilities and industry, those facilities, when they're producing antibiotics, often will have waste discharges that often pharmaceuticals are not one of these permitted, regulated effluent points that we need to think about. So we are often concerned about where that effluent goes. The other thing is in plant agriculture, for growing things, antibiotics are actually often used as pesticides. So especially when you're talking about things like a kind of a fungus or a rot, we will use antifungals or antibiotics to help reduce or improve agricultural yields. And each of these spheres, like we don't live in silos, right? I think a lot of times we've been thinking about research as happening in these distinct boxes, but really what happens is, right, those cows and those pigs, they go on those fields in the environment. Or often you have plant agriculture happening next to animal agriculture. or as an individual, we swim in places where wastewater treatment plant water is discharged. And we use increasingly for water reuse, we use treated wastewater to irrigate plant things. We have to think about these things all like a system. And I'm not the first one to think about this. So the World Health Organization and the United Nations Environmental Program, we're starting to think about things in this one health framework with the idea that in order to maintain human health, and this applies to not just antibiotic resistance, but zoological disease as well, we need to make sure that we're looking at healthy animals and healthy ecosystems, because we know healthy ecosystems can really help to buffer our exposure to the environment. So here's probably, I was excited about this figure, but it's probably a bit too much, where we tried to visualize all these different contributors to the environment and what that might be. If it is antibiotics, if it's antibiotic resistant bacteria or antibiotic resistance genes. And we see that there's actually a lot of inputs and there's also a lot of outputs. So wastewater treatment or agricultural wastewater might discharge to the environment, but downstream, human population might use that for drinking water, or it might get used for irrigation water. And so first, thinking about this as a big system, we can start to think about how are they connected and what are our critical interventions. So a big scary report that came out last week, I'm a fun person at parties, always looking for these scary reports, is this new study that came out last week from the UK with the idea that superbugs will cost more than $2 trillion a year by 2050. And if it continues to go unchecked. And two of the actions that these researchers reported is that we need to develop new antibiotics, so we have new solutions, but they say that they assume that drug resistance emerged at the same rate. So the question becomes, can we slow the resistance of the emergence of antibiotic resistance? And I'm specifically, you guys might have figured this out because you're in the wastewater field, going to talk about critical infrastructure interventions that we can employ in water reuse and wastewater treatment that will help to decrease the evolution and emergence of antibiotic resistant bacteria. So we still have to think about this, and I think many of you are familiar with this idea that wastewater treatment serves as a meeting point, right? It's not a static process. I think often when you talk to people who don't know much about wastewater, I don't know if they think of it like a light bulb or what it is, but you turn on the treatment plant and there it goes. Anyone who's in wastewater knows that it's a bit more complicated than that. And we have to realize that in wastewater, at the beginning of treatment, in the sewer pipes, or even as we go, wastewater is going to be unique to different contributors, right? So you can imagine that hospitals are maybe going to have more antibiotics in their wastewater because of the prevalence of people in the hospital who might be needing critical care. They also might have more pharmaceuticals and personal care products. Homes, though, something I'm constantly talking to my students about is actually whenever I've been prescribed antibiotics and I wasn't in the hospital, they gave me a prescription, right? And I filled it at the pharmacy, and I actually took those antibiotics at home. So when we think about antibiotics and where are they present, we have to realize that the wastewater coming from our homes actually might contain a lot of antibiotics as well. And I want to back up a second because I think I missed this and I want to make sure. When I'm talking about antibiotics in the waste stream, I think it's a little disconnect from when I'm saying you take the antibiotics and somehow they end up in the wastewater. About 80% of the antibiotics that we take as medicine, so we need them to stay active right when they travel through your body and they treat those infections. Most of those antibiotics are going to stay intact. And so they're going to get excreted into your wastewater, which is why I'm talking about antibiotic concentrations in the waste stream. The other thing, though, that happens when you take antibiotics is that antibiotic resistant bacteria, and it's not by design, it's just that they happen to live maybe in your gut or on your body, they might survive. And so then when you use the bathroom, they also enter the waste stream. So what we see is that wastewater treatment can often serve as this confluence points, right? All these different sources are mixing together, and bacteria that didn't previously have antibiotic resistance, they might actually come into contact with bacteria that do have antibiotic resistance. And specifically what we're finding is that resistant bacteria seem better able to survive in the harsh conditions of a sewer, which is honestly a little bit of a bummer. It's a little scary. I kind of wish those were the weak ones, but the clinical bacteria actually might survive treatment or obtain new genes throughout the sewer or the treatment train. We're going to look at some data kind of as we keep going. But I wanted to also talk about, all right, we're going to talk about how we can measure antibiotic resistant bacteria, because, okay, we know it's a problem. We know it's relevant to wastewater because we're serving as this big confluence point of everyone taking antibiotics and, using the facilities, as it were. So we need to now figure out, how do we measure this? And there's a few different methods. And so I want to make sure we're all on the same page. We can use culture methods. So this is probably what maybe you all are most familiar with, because it's how we do fecal coliform testing. That's a standard protocol, but it's essentially the same idea, that you can take wastewater that you collect either, from wherever in treatment, it'll change the volume, but take your wastewater. And then in this case, you would use media that has with and without antibiotics. So you can get the percentage that are resistant within your waste stream. A downside of culture-based approaches is developing those culture methods is really challenging. So those fecal coliform tests that we use and we regulate on, those took a long time for the EPA to come up with standard methods. It wasn't like we woke up one day and said, here we go, we're ready. And so most of the cultural bacteria that we have methods for are really focusing on those fecal bacteria when we think about environmental samples. The clinical samples that we're used to taking and measuring all these things, those aren't designed for wastewater, and wastewater is a bit different from a human clinical sample, and it can mean that there's background contamination. And by contamination, I just mean the things that grow aren't what you want to grow and what we want to count. The other way we can do it is a gene-based test. So these are often used when we're trying to assess environmental antibiotic resistance, because what they let us do, so cultural bacteria is only going to let you count the living cells, which is good. We want the living cells, but it will only let you count the ones that you have methods to target. And many of our wastewater bacteria specifically, we just don't know how to culture, right? And we don't have good methods. We call them unculturable. I like to think of them as not culturable yet, but we still need a way to measure these genes. And so gene-based assessment can allow us to identify total antibiotic resistance genes across the microbial community. And then we can gain insight into any mobile genetic elements. And that's important because one of the elements in antibiotic resistance that's really challenging, and why we think about superbugs, is because often they can survive many different antibiotics. And this is because bacteria can actually transfer genes. Like you could transfer genes to your friend. It doesn't just have to be to your children. So they can transfer genes, we call it horizontal gene transfer, to each other. And this gene-based analysis lets us see it. You can do two different ways. So you can target one bacteria at a time, or you can do kind of a shotgun approach where you look for everything. But the real drawback in gene-based analysis is we can't distinguish live versus dead. So what this might look like kind of as a workflow is for your sample collection for culturing, you would do filter concentration, and then you could culture for specific organisms or traits. I want to make sure you all can see this. You culture for specific organisms or traits. For gene-based analysis, we would first do a DNA extraction, and then we can amplify just those specific genes at a time. But as I said, these genes are constantly evolving. So sometimes you actually want to do what we call a shotgun approach, which means that you would get all of what's happening. And that's obviously a bit more data heavy. So it's kind of really still a research method, but it does let you get an idea for, is there anything new going on here? And that is incredibly valuable for thinking about the evolution of resistant genes. So for a sequencing workflow, it would be similar. You do DNA extraction, and then you would do sequencing. And it would let you annotate to existing databases. So see if there's any, looking at all the antibiotic resistant genes at once. It can also, through metagenomic assembly, start to try to identify, do we know which bacteria and which gene? Okay, so that's a lot of methods. We'll now start to think about exactly kind of what we're more familiar with wastewater treatment processes and exactly how do they handle antibiotic resistant bacteria. So this is from a project I did quite a bit ago at Virginia Tech, working with a few different full-scale utilities. And what we actually started with is looking at treatment technologies and water reuse. So I'm going to start from the jump here. I know that many people in the finance network are coming from small systems. And quite honestly, we don't have a lot of data from small systems. I'm working on it this summer. It's going to take my grad student a little bit of time. So I'm going to start with kind of what we know, looking at these really advanced treatment processes. And then I really want to challenge you all. We can keep thinking going forward, how could we assess this in smaller systems that maybe don't have like these crazy treatment trains? So if we're thinking about, in this study, which is published, so you guys can look it up, we looked at 2 water reuse systems, and they're doing 2 different objectives. So we had one, this top one here, that is using wastewater. They're treating it, but they're only treating it for non-potable reuse. So they have a wastewater secondary clarifier, then they have a denitrification filtration system, and then they have chlorination and chlorination storage, and then they give it to a municipal network for purple pipes. This one's in Florida, so they use the purple pipe system, and so consumers know that this is non-potable, and it's really intended just to be used for irrigation. We have another system that's in Virginia, and this is actually producing potable drinking water at the end. They are not using it to distribute as drinking water. They are using it for groundwater recharge. So this is a secondary clarifier, and then they actually looks like they just took a wastewater treatment plant and they kind of attached a drinking water treatment plant on the other end. So they've got coagulation and flocculation. They actually add some chloramine early in the system because it improved the ozonation. They've got biological activated carbon filters and granular activated carbon filters, and then UV disinfection, which actually will get routed to discharge. So in this study, what we did is we sampled 2 different treatment plants. And we sampled them only 4 times a year because that's what we were limited to kind of with the money that we had. And we did culture methods and metagenomics. So just as a refresher, what that looked like was then we did that 2-liter filter concentration, we did DNA extraction, and then we aligned to our databases. And then we actually tried to target all 6 of the CDC critical pathogens of concern for antibiotic resistance. And we'll talk a little bit how that went, kind of getting at what I was teasing with these culture methods that were challenging. So looking at just the culture methods initially, and I realized maybe for you, these are a little bigger. For me, it's a little small. That's okay. So if we look at, we started really ambitiously looking at these 6 target organisms. However, 4 of those organisms didn't have EPA standard methods. And it shows, honestly, we did a lot of work to get these different bacteria to grow. But then when we checked their genes to see if what we were targeting, so they looked right on the plate, we were counting them. Then we checked if they were what we were looking for, and they actually weren't. So you can see here, a lot of these results we didn't get to do much with because only 3% of our Acinetobacter baumannii turned out to be what we thought it was. 15% of our Klebsiella pneumoniae was on target, and so on for these different ones. And this was a bummer, like as we were doing the study, but I really want to highlight, I think it really shows that if we want to look for these really resistant bacteria in wastewater or complicated systems, we need better methods. And methods development is maybe not the most fun thing in the world, but it is really needed. But if we look at the 2 culture methods that we did, I realize my head's in front of it, so I'll slide over. We'd look at the 2 organisms that we have EPA methods for. So that would be our Enterococcus species and our E. coli. These are both considered fecal coliforms. If you were looking at them, they are both in the fecal coliform group. What we see, if we start with just our potable reuse facility, is that we're looking at our total bacteria. So this is showing that, yes, wastewater treatment is effective at removing bacteria. If we just look at our totals and we look at them across all of our plants, look, we're getting huge reductions in bacteria as we go through wastewater treatment. But if we look at our resistant bacteria, what we look for as researchers and what we're a little concerned with is you see at the beginning, especially this top plant, maybe there's 4 log orders magnitude between our total bacteria and our resistant bacteria. And we see, initially there's quite a big split between total and resistant. However, as we go through the treatment train, we see that those lines tend to get closer together. And that means to us that we are actually enriching for these bacteria at some stage. So specifically, you can see it here. By the time we're getting to secondary clarification on our Enterococcus, most of the bacteria that we were counting were actually resistant. So from a wastewater treatment process, that kind of worries us, right? Because we want to be protecting public health through the water that we discharge. And we don't want to be enriching for resistant bacteria. That's not the goal. So while we are getting, we did still get below the limit of detection for quite a few of these. I do want to point out, especially in our one with the distribution system, when we sampled right after treatment, we met the EPA standard. But what we saw when we looked in the distribution system was actually that there was quite a bit of recovery, and that the recovery of E. coli, they were actually mostly resistant. So we're not regaining that split as they recover. And so here's those a little bit bigger. We can kind of talk through them a little bit more. If we just look at the non-potable plants, so I want to just point out for the Enterococcus that we are seeing that increase across our facility and when we look in the distribution system. But I do want to highlight wastewater treatment is obviously removing a huge amount of bacteria. So it's not like we're not doing our jobs. It's just thinking of ways, do we need to be improving to face these new challenges that are emerging? So the next phase, I mentioned this, and this is a lot. So I know it's overwhelming. Just kind of take a breath and I will walk us through it. Do not worry. So if we think about, we looked at that potable reuse facility. We did culture methods, which we looked at and which didn't maybe go as well as we wanted. They worked well for E. coli and Enterococcus. But if we do sequencing, it lets us see all of the antibiotic resistance genes at once. So for this facility, in our influent, our primary effluent, we measure, you can kind of think about it like, this certain proportion of our total community was carrying antibiotic resistance genes. And as we go through the treatment plant, that's going down. So we're still seeing removals of bacteria through this other method. However, what we see and what was kind of alarming to us as researchers is that there's some stages and some treatment processes that are causing things, once they go down, to come back up again. So specifically, one of the ones we looked at was ozonation. And so what does that mean, right? So there's two different theories, right? Are we removing so many bacteria that if we think about enriching for resistant bacteria, that there's just so few there, and then the ones that remain are resistant, but we've removed most of them? That's possible. What we think might be happening is that the bacteria that are resistant to, that are drug resistant, actually seem more equipped to survive these really harsh disinfection processes, which is an interesting engineering challenge. I used to think of it like an engineering challenge. And then as we go through for BAC, biological activated carbon filters and granular activated carbon filters in UV, things were pretty stable. But we did again see a really big enrichment in chlorination. And the other thing that we see that was interesting is if you look at the makeup of the colors of these bars, that they do shift a little bit as you go through the treatment process. So we start with kind of one makeup, maybe it's these yellows, browns, blues, and greens. But then by the time we get to the end, if we're really looking, we have like this new color blue that's emerged. The yellow has kind of gone away. I don't know where the brown went. The makeup of these genes has really changed. And then in this facility, we also monitored those groundwater wells. And so my goal is not to scare you. My goal is to teach you about challenges that we're thinking about in research, is that we did see, if we looked at the 3-day groundwater well and the 6-month groundwater well, those actually were still lower than the background well that we were monitoring, which is reassuring. We suspect this is because of dilution, right? Because this was a very small facility discharging a very small stream to the groundwater, but we are watching that as we keep going, and as they add more, they increase the volume that they're recharging. So if we look in the non-potable reuse system, what we see is that we're looking at these metagenomes, that we've got our influent and our primary. So this is the non-potable system. So it doesn't have as many treatment processes. And so initially though, if we look at our influent here and we compare it to our primary clarification, we've got those same makeup that we saw before, which really as an engineer is kind of reassuring to me. It means if we compare a Virginia plant and a Florida plant, those influents look fairly similar. And actually these different treatment processes are performing fairly similarly, which if we think about designing new solutions is really reassuring. However, one of the challenges that we noticed is that after denitrification, this chlorination step again, when we measured it in a different facility, it was also causing an increase in antibiotic resistance genes. So this was a non-potable reuse system that's putting it into a distribution network, which is obviously not most wastewater treatment facilities. But what we see is that, so there was a slight increase again, this different facility. And also, we look again at those makeup of those bars, at the different colors, it looks like chlorination is really changing the makeup of the antibiotic resistant bacteria that are there. And that's not going back to normal, really, or back to the influent, which isn't really normal. It's kind of gross, but it's not really changing once we leave chlorination. So we have a chlorination storage, and then we get to the distribution system, we see significant increases again in resistant bacteria, which is quite alarming if we think about that that's probably the closest to the consumer. So kind of a take home here is that our chlorination, it increased. It wasn't significant in this plant as it was in the other one, but it was still slightly there. But that we do need to monitor the reuse distribution systems. So I know this one, again, I'm giving you guys a lot. I'm trying to kind of show you where we are as researchers. If we look at those, we've looked at those as stacked bar charts. And those stacked bar charts are kind of fun because we have all those different colors. And you can see those profile changes over time. So profile just meaning the different makeup of the bars. But if we look at, but you couldn't really tell if we were comparing just one, like just that blue bar or just that pink bar, what was happening to it throughout. So what we did here is each of those colors is now a row. So you could see if different treatment systems, this is just on the reuse water side, were enriching or not enriching for different types of resistance. And what we want to see is across both facilities, are we seeing any similarities or differences really? So we see honestly that it's mixed, but most resistance genes, this is a take home, are being reduced, especially when we have this potable reuse effluent. So the potable reuse does seem very, very effective at removing antibiotic resistance genes. However, we do have this other category. And so most of those are actually antimicrobials. And antimicrobials are these chemicals that might be present in your dish soaps or in, I don't know if you guys have get those ads for those copper socks, but they have all these different antimicrobial properties that we put in consumer products that aren't antibiotics. We do see that those are still persisting. And that's one made us want to look into that more. If we look at our non-potable reuse system, it's a little bit less of a fuzzy story, warm and fuzzy story. We again see that the other category, if we look in the distribution system, is increasing for that other, but we also see it's increasing for a few other classes. This might be just because there's less treatment processes, so there's less options to remove things, but it is something we want to watch, and it's something that we're continuing to do research on here at Michigan Tech to see if there's different types of disinfectant we could use in these systems to keep those from regrowing or to not enrich for it as much. So just looking at, we can think about antibiotic resistant bacteria and genes. They're not all created equal. So the CDC publishes a list of those critical antibiotic resistant bacteria, and those critical ones really do mean we don't have a lot of treatment interventions. And we can think about those as human health relevant genes. However, there are some antibiotics and antibiotic resistant bacteria that are really resistant to antibiotics that we don't really use as much in human medicine. So there's some antibiotics, we don't use them because they don't work anymore, which is unfortunately means that resistance has kind of evolved. But what we wanted to do here is look at if we just focused on the human health genes or those environmental genes, do we see differences, right? Like we really want to prioritize those human health relevant genes. So what we do see if we look at, this is kind of a risk score metric, we get significant decreases in every category during wastewater treatment. Wastewater treatment is effectively removing resistant bacteria. However, especially in those reuse distribution chains, we do see increases still in human health relevant resistant genes. For the ecological genes, we see similar trends for our clinically relevant and our ecologically relevant genes. But if we look at our ecological genes in our non-potable system, it again really looks like that distribution system is allowing for regrowth. Also, what I wanted to highlight here is that, earlier we were talking about, are there problem spots in our treatment trains that are causing for enrichment of these resistant bacteria? And two that really came up that we talked about were ozonation and chlorination. So when we look at this at a finer scale, what we want to see is, so what we see here, if we just look at that increase in ozonation, it doesn't look like that's happening because of human health relevant resistance genes increasing. It looks like that's happening because of environmental relevant resistance genes. So this doesn't mean it's not important. It doesn't mean we shouldn't try to figure out what's going on, but it does mean maybe we can breathe a little bit of a sigh of relief that this is, it's still happening. We should still figure it out, because what we don't want is maybe a clinically relevant bacteria could have both of those genes at once. That can happen. And it's kind of the scarier superbugs that are out there, but it does show us kind of where those resistances are coming from. So just kind of a little highlight is that the increases in ozonation really seem to be because of ecological resistance genes. So that was really water reuse. And I know you all are mostly wastewater treatment operators, so I wanted to talk through a study that came out just last year that looked at different treatment plants across the world. And so we can think about what's going on in terms of those resistances and how are they reducing or enriching. So what we do when we think about that, this is the same kind of plot that we saw earlier. So in this study, what they did, or what I did, I helped with this one as well, is we sampled in 8 different countries. So across, we have different, most of them we tried to have the same treatment technologies. So we're comparing apples to apples. They all had primary and secondary clarification. I think they differed a little bit in their solids handling and their disinfectants. But if we here we were looking at those influent, effluent. We're just comparing how does wastewater treatment do if we just compare influent to effluent across those different drug classes. So we sampled in the US, that was in Virginia, we sampled in Sweden, we sampled in Switzerland. So these are 2 places often with antibiotic resistance. Specifically, we think about how the different regulations for antibiotics in different countries can cause there to be different levels. So the US, we have you need to have a prescription to get an antibiotic. And as of 2018, animals also need a prescription to get an antibiotic. And I do want to point out that is a change, right? Implementing those more stringent requirements to get antibiotics is how we think about ways we can reduce the amount of antibiotics that are used, especially if they don't need to be used. Like if it is a virus or we're using antibiotics for growth promotion and not for maintaining animal health. So we sampled in Sweden and Switzerland, and these are 2 countries that are really high income countries. They're in the global north, so they're going to have colder seasons, and they have really stringent effluent standards. Their standards are probably more stringent than ours. And then we also wanted to sample in a few areas that are going to be a bit different from the US. So Switzerland and Sweden, like the US, require prescriptions for all these different things. Sweden actually stopped the use of antibiotics in animals pretty early, around 2007. So we were wondering, does that mean they have less resistance overall in their population? Then we also sampled in India, Hong Kong, and the Philippines. So India is a warmer country. It's a less socioeconomic. They're a middle income country, so they don't have maybe as many resources to manage their wastewater quite as effectively. And a notable cultural difference in India is that antibiotics are not prescription drugs. So often you can just walk into the pharmacy and get an antibiotic. And that we were really wondering, does that mean that the wastewater is going to have more antibiotic resistance in it? And does the treatment still work as well? That's a big question. Hong Kong is a high income country that does, I think, require prescriptions. But the Philippines is similar maybe to India in that it doesn't require a prescription for antibiotics. So we're comparing a lot of these different factors when we think about these 8 different countries. So looking again, this is just a reminder, we're comparing influent to effluent. So we see for our different antibiotics, what we see is if we have a decrease, that's green, and an increase is red. That again, we have some overall trends with a few outliers. So like one I want to point out is tetracycline resistance. So tetracycline is often used in animal agriculture. It's more of an environmentally relevant drug class, but every facility that we sampled was really good at removing tetracycline resistance genes. However, most facilities were really not good at removing trimethoprim resistance genes, which is another drug class. That one's actually a bit more clinically relevant. Similarly, for rifamycin and sulfonamide, it looked like the India plants maybe were very effective at removing them. And we have to think about why that might be. Like maybe they started higher. And so then at the end, it is a reduction, but it's because we started initially higher. But most of the higher income facilities, so the facilities located in higher income countries, saw that they were increasing for those resistant bacteria. As we go through other different drug classes, like things like peptides, phenocols, quinolones, all of those were really effectively removed during treatment. And just remembering that these are maybe more conventional treatment processes than those water reuse systems that we talked about. And then thinking about multi-drug resistance genes or specifically MLS resistance genes, those were all effectively removed. So I want us to really think about this. Like we looked at the most extreme treatment examples with water reuse, and we saw that for the most part, we can get reductions, but it seems like those distribution systems are going to cause increases. If we look at a conventional facility, you still get reductions. The question, and this is the big research question that's still going on, is what reduction do we need? So the reason that we set certain effluent standards for like fecal coliforms and things like that is because we want to preserve the quality of the surface water, and we ultimately want to reduce the risk that someone downstream of a wastewater discharge could get sick with a bacteria coming from a wastewater treatment plant. That's really our goal. Because we don't want people downstream to get sick because we didn't remove enough. So with antibiotic resistant bacteria, something we're thinking a lot about is like, which genes should we prioritize? I think instinctively we want to prioritize those clinically relevant resistance genes because we really don't want someone to be swimming in water downstream and to potentially, maybe they swallow a little bit of water, maybe they're splashing a lot because they're a kid. We don't want them to accidentally get exposed and to potentially have a resistant infection. So in this bottom panel, we look at specific resistance genes, and these are resistance genes, but honestly, that are pretty scary. These are the resistance genes that we don't often have treatment for if you get them in the hospital. And we're again looking for those if they increased or decreased throughout wastewater treatment. So most of them, you'll actually see these dots are pretty small. That means that there was no change. So we didn't enrich for them, but we didn't decrease them. And that's kind of a problem, right? We think we would want most of them to decrease. Certain ones like blah OXA, for the most part, we got really big reductions, which is great. But other ones, so blah carb, CARB, it is a clinically relevant one. It's kind of a newer one, and it looks like it was increasing through treatment. So when we think about antibiotic resistance, I think something that's complicated is we can't think about it all like one thing. It's actually made up of many different drug classes, and those drug classes can be conveying resistant to different types of drug resistance. And maybe they're not all the highest priority. Those clinically relevant ones maybe are our highest priority, and those ecologically relevant ones, well, we would like to treat them, are just a little bit lower priority than what we're concerned about. And then once we get within those drug classes, we have to look at individual genes. And I know it's a lot. And I think on the water utility side, what I want you guys, or the wastewater utility side, to take home is that we are getting reductions. And something like I mentioned, a lot of you all are coming from smaller facilities. I would like us to get better methods and better assessments for how do small facilities handle antibiotic resistant bacteria. There's just not a lot of papers out there. Michigan Tech is really unique in that we have a research facility in a small rural area that can ask that question. And we are. It's just going to take us a little bit because I'm relatively new here. But we're kind of getting close to where I'm going to ask you all for some questions. So first, I think it's really important to understand where we get money for these types of studies. We understand where this money that these agencies are getting goes. So this money was a lot of different funding sources kind of all compiled into one. We received funding from the Center for Disease Control and Prevention. They obviously monitor most antibiotic resistance in healthcare systems, but they're also really concerned for these environmental exposures, if people could be getting sick from effluent. It was also funded by the USDA. So USDA does a lot to mitigate antibiotic resistance, especially in water reuse systems, because there's a lot of non-potable reuse that's happening. We want to make sure that we're doing it safely. We also got funding from the Water Research Foundation. So the Water Research Foundation is actually a utility sponsored organization, so utilities can join, and they help fund utility topics that utilities are interested in specifically. So they're an industry sponsor. And then these are more people who paid my salary throughout, but I did a postdoc at the National Institute for Standards, so that's helping us develop better methods to detect antibiotic resistance. Virginia Tech, and then the National Academies of Science, Engineering, and Medicine also helped to fund me doing this work. So I know it's a little bit early, but I'm hoping we have a robust discussion. And if we don't, I can talk about this for forever. So kind of at that, I would like to ask if anyone has any questions. And I think Lori is going to really help me manage the Q&A, but they should pop up pretty soon.
Yep, right now we don't have any in the queue yet, but as soon as we have them streaming in, I will throw them at you.
All right, and yeah, feel free to drop any questions you have in the Q&A. I'm sure there's things I didn't cover, or there's things we can talk about even more.
Sure, we have one here. Have you tested antibiotics in waste activated sludge? And presumably antibiotics tend to accumulate in waste activated sludge. Is there any study ozone destruction in waste activated sludge antibiotics?
Yeah, so part of this study, we didn't talk about it a lot, quite honestly, because it's not my expertise. I'm more of a resistance gene person and not antibiotic concentration itself. We did measure antibiotics in activated sludge, not specifically just the waste activated sludge stream. Now, what we do see is that, one, there are detectable concentrations of antibiotics throughout the waste stream. We think that they are helping to select for these resistant bacteria, but they're at really low concentrations. And so it can be hard to measure that low, quite honestly, because we're talking about so much dilution. But yeah, antibiotics, they don't accumulate maybe as much as you would think, because different bacteria actually will, so these pharmaceuticals and antibiotics, if you are a bacteria that's resistant to them, there's a lot of energy contained in these complex pharmaceutical chemistries. They can actually start to break them down a little bit. So usually we also want to look for a lot of the antibiotic metabolites, because they will start to break down in the wastewater stream. I haven't specifically looked at ozone destruction for waste activated sludge antibiotics, but it does sound like a really interesting study, and I'm always willing to talk about it more.
Okay, thank you for that. Our next question, how different types of chlorination, chlorine gas or chloramine, et cetera, affect antibiotic resistance?
Yeah, that's a great question. And I feel like you guys are going to have these awesome questions. And honestly, this field and this area of study, we've honestly been more in these broad strokes stage. So I think we're finally at the point where we've established that this is a problem we need to look into. But I really haven't seen a lot of studies, especially at the full scale, right? So one facility is probably only doing one type of chlorination. There's not very many facilities that have it set up to do different types. So we can compare different technologies specifically on one wastewater. But if we want to look at the full scale, it's really hard to compare multiple different technologies because they're just not implemented. So we're working in our lab to set up a disinfection rig that we can try many different disinfectants on one common wastewater, because we saw that each wastewater, it can be a little bit different at the beginning, and you don't want what's starting to affect how you think of something as performing. So I don't have a better question. I can't tell you which one's better overall. I can say that we are a little bit concerned about chlorination specifically, but we're not at the stage of having guidelines. So we definitely know that chlorination is effective at removing total bacteria, and we don't want to get away from that message, right? Disinfection is doing really critical work. Now we're starting to maybe dig into it a little more to say which type of disinfection is really going to be the best guidance going forward.
Great, thank you. We're still waiting for any additional questions. I don't know if you have more that you want to elaborate on, Ishi. Otherwise, we can talk about like how they can get their certificates. So it's up to you.
Yeah, I can talk through a couple, I guess, things that I talk about a lot with people that are interested in, and we can maybe food for thought. A lot of this is food for thought. So when we think about antibiotic resistance, I think we hopefully did a good job of connecting the role that wastewater can play. But we didn't talk a lot about the role of different bacteria acquiring these different genes. So that could be a whole nother talk, just looking at this enrichment process. But we have to fundamentally remember, like, wastewater has a critical role. We don't want to disrupt those processes too much. What we really are looking at in my lab is, are there specialized, maybe an add-on process? I know no one wants to think about that, but we're on the research side. We'll just see if it works first, and then we'll talk. Or are there ways we can maybe specifically use these biotechnology tools to target specific genes that we have? I think other things that we think about, is really, I think some of the questions I've really got is, you're measuring genes, you're not measuring, cultural bacteria. How real is this problem? And unfortunately, I guess now we do have a few studies that are out there that actually have clinical isolate matches of bacteria surviving fully wastewater treatment and downstream. So that's really, I guess, a lot of the motivation. I don't know, Lori, I see a new one that came in the chat. Do you want to read that one?
Sure, absolutely.
Great, thank you.
How often would you test for antibiotics in wastewater for rural areas? Just testing quarterly, or should more testing be done? What we have noticed in our rural area is an increase in uranium.
Yeah, so I think we're not at the stage, and unfortunately, I understand that wastewater utilities, you guys never have just tons of extra money laying around to test for things. And so testing for antibiotics in the wastewater, I think one of the things we're thinking about is, if you have specific, known industry contributors to your network, we could envision a future where we make sure that those antibiotics that they're contributing are getting reduced before they enter the municipal sewer. But we don't yet have targeted, like, which treatment process that you should do to remove specific antibiotics. So I don't know that we're at the stage of having everyone go and test. I mean, from a data point of view, I love this idea, and but I would really recommend, if you were going to do that, test kind of the effluent, or actually in maybe just after activated sludge, to see what's getting through your most biologically enriched process. Testing in your influent, you will see antibiotics. I'm actually pretty confident about that, but we don't necessarily have targeted removals yet. I do want to point out, so I think the increase in uranium is really interesting. That might be one, honestly. I mean, in the rural area where I'm at, some of the different groundwaters can actually have some uranium, which does involve, and it would be groundwater, and then people excreting it into wastewater, and then you're seeing that increase. So that would be maybe kind of a groundwater contamination issue, or maybe it's naturally occurring. Something that we, trying to remember where I was. I lost my train of thought a little bit there, guys. But when we think about how we want to test for antibiotics, or what does this future look like, I just think we don't have exact guidance yet on quarterly. Different groups that have found it, we do see that it's fairly consistent over time. But the real challenge, like I mentioned, is kind of, I guess it's a little bit similar to the PFAS challenge that a lot of us have been hearing about, is it's not just like we have one point source. Like, when we have one source that's industry, or we have one source, then we can usually, have that source reduce things before it's discharged. And I do think for hospitals that might be a direction we go, because they are having a lot more, that's where the clinical bacteria are. It's also where a lot of pharmaceuticals are used. But for other types of people, like I mentioned, there's antibiotics are used similar to PFAS in people's homes. We can't, obviously, like, wastewater treatment is the central way to treat that. So we don't have a solution yet on exactly testing, but the studies I've seen has seen that it's fairly consistent over time. So you might be able to, quarterly would probably get you an idea of what do we have, and then you can start to think about what could you do. And then so that's I don't know if we have any more questions. Or Lori, you want to talk about how people can get their certificates?
Sure. How about if any more questions come in, as they come in, we'll sort of leave it open. And we'll be here for another 5 minutes at least, right? So if something strikes you, please do go ahead and put it in the Q&A or into the chat. In the meantime, I am going to go ahead and put the information that you need to get that certificate into the chat. And if you have any technical issues with this, we will be here for support or questions. And you should see that shortly. But please do go ahead and click on that link, complete the evaluation questions. And if you run into any trouble, even after today, just shoot us an email at that CTT email address there.
All right, and then I just want to say again, my email's up on the screen. If you want to reach out or anything, I'm happy to talk. I'm hoping to equip my students and do research that's really relevant for utilities. So as you guys have questions in the future, maybe feel free to shoot me an email.
Thank you, Ishi, so much for all the great information. And if we have any more questions come up, I'll let you know. If they come up before we close today in just a few minutes, I will forward them to you as well. But thank you so much for joining us.
Sounds great. Yeah, and I see in the chat there's some conversation. We didn't start to talk about biosolids. I think similar to what this person is mentioning is we've also seen that most antibiotic resistant bacteria, we have to remember if they're removed in the liquid fraction, so that wastewater effluent, that means they went somewhere, and a lot of times they're going into the biosolids. But biosolids treatment might be effective at removing them. So we're always balancing those different treatment processes, and it's a lot to fit in one presentation. I don't want to scare anyone, Jen, but I'm glad you liked it.
Well, thank you too to everyone for your attendance again today. We're so glad that you could join us. We'll be here for just a couple more minutes. If something does strike you and you have a question, go ahead and put it in the chat. We will be here. Otherwise, you are welcome to exit the webinar if you're ready to do so.